So, you've probably heard that our DNA is the blueprint for life, right? It gets copied into a message, this thing called mRNA. But how does a message actually become a real physical machine inside our bodies? Well, that's what we're going to do today. We're stepping inside the cell's protein factory to see exactly how it's done.
And that really is the big question, isn't it? I mean, how does a simple code, just a string of 4 letters, A, U, G, and C, turn into something real? Something you can actually use, like an enzyme that breaks down your lunch or the hemoglobin carrying oxygen through your veins right now. It's, it's like magic, the ultimate act of biological creation. Okay, to really wrap our heads around this whole thing, let's think about it a different way. Imagine a huge, bustling construction site, but shrunk down to the molecular level. We've got blueprints, a build crew, delivery trucks, the whole shebang. We're going to see how a complex machine is built right from scratch. All right, here's the plan for our project today. First up, code to construction, how the blueprint gets to the site. Then we'll meet the build crew, the key players. After that, it's the assembly line, watching it all happen. Then, from chain to machine, the final touches. And we'll finish off with a quick test your knowledge. Simple enough? Let's get to it. So, first things first, you can't build anything without a plan, right? In the cell, that plan, our blueprint, has just been freshly printed in a process called transcription. Now it's time to get that blueprint from the main office to the actual building site where all the action is. And here's the key thing to remember. The original master plans, that's our DNA, they're locked away for safekeeping in the nucleus. Think of it like the head architect's office. You don't take the originals to the muddy construction site. Instead, you make a copy, a blueprint. That's our mRNA. And that copy gets sent out of the nucleus into the cytoplasm. And the cytoplasm, that's our bustling, noisy construction site. This is where it all goes down. Okay, so we're at the construction site, but you can't build anything without workers and machinery, right? Well, our cellular construction site has an amazing, highly specialized crew, and every single member has a critical job to do. So, let's meet them. First, of course, is the blueprint itself, messenger RNA, or just mRNA for short. It's just a single strand carrying the instructions copied directly from a gene. And the language it uses is super simple, just 4 letters. But believe me, the order of those letters is everything. This is our guide for the whole project. Next up, meet the factory. The general contractor, the assembly machine, this is the ribosome. And it's an incredible piece of molecular machinery. It's actually made of 2 parts, a big one and a small one, that come together and literally clamp down onto our mRNA blueprint. Its entire job is to read that code step by step and build the protein. It is the absolute heart of our construction site. Yeah, check this out. You can see it perfectly here. You've got the small subunit down below and the big one on top. And look how the mRNA blueprint just threads right through the middle, kind of like an old school ticker tape. It's this perfect snug fit that makes everything work. And last but not least, we have our delivery trucks. These are the transfer RNA molecules, or tRNA. And honestly, these guys are the unsung heroes of the whole operation. Each tRNA is basically a specialized truck that can only carry one type of cargo, a specific building block called an amino acid. And how does it know where to go? Well, it has its own 3-letter license plate called an anticodon that has to perfectly match a 3-letter address on the mRNA blueprint. No match, no delivery. All right, the crew is assembled, the blueprint is unrolled. It is time to fire up this assembly line and actually build something. This whole process is called translation, and it happens in 3 main stages. Think of it this way. First, there's initiation. That's like laying the foundation and getting everything set up. Then comes elongation. This is the main build, adding the bricks floor by floor, and finally, termination, finishing the job, cleaning up, and packing everything away. Let's break them down. Okay, so initiation. It all starts when the small part of our ribosome factory latches onto the mRNA blueprint. It kind of slides along, scanning, until it finds a very specific 3-letter code, AUG. That's the universal signal for start building here. As soon as it finds that, the very first tRNA truck pulls in, carrying its specific amino acid, methionine. And only then does the big ribosome subunit come and clamp down on top of everything. Boom, the factory is officially assembled and open for business. Now for the main event, elongation. This is where the real work gets done. The ribosome starts chugging down the mRNA track, reading the blueprint 3 letters at a time. Each 3-letter word is called a codon, and for every single codon it reads, the exact right tRNA truck zips in, delivering its amino acid. Then the ribosome, like a master welder, forges a super strong bond, a peptide bond linking that new amino acid onto the growing chain. And this gives us a fantastic peek inside the factory. See those 2 main slots inside the ribosome? We call them the A site and the P site. Think of A for arrival. That's the loading dock where the new tRNA truck pulls in. The P site is where the growing protein chain is being held. So watch the rhythm. A new tRNA lands in the A site. The ribosome snips the chain off the tRNA in the P site and attaches it to the new amino acid in the A site. Then the whole machine scoots over one spot. The now empty tRNA gets kicked out and the A site is free for the next delivery. It's just this beautiful, efficient, repeatable cycle.
Seriously, just imagine for a second you're one of these tRNA molecules. Your whole life is this one job. You grab your specific amino acid, you hang around waiting for your 3-letter code to be called out on the assembly line, and when it is, bam, you zip into that A site, drop off your cargo, and you're immediately ejected off to find another amino acid and do it all over again. It is a non-stop, super precise delivery service.
And this just goes on, codon by codon, amino acid by amino acid, building this long chain. Until the ribosome hits a roadblock. It reads one of 3 special stop codons. Now, these codes don't call for a delivery truck. Instead, they're a signal for a specialist to come in, a protein called a release factor. It swoops into the A site and acts like a molecular pair of scissors, just snipping the finished protein chain free. And that's it. Job's done. The whole factory falls apart, the ribosome subunits split, and the mRNA blueprint is released, maybe to be used again to build another protein. So there we have it. We've built this long chain of amino acids, a polypeptide. The construction project is finished. Or is it? Actually, no, not even close. In fact, maybe the most critical step is what happens next. Because right now, all we have is a long, floppy noodle of amino acids. And on its own, it's completely useless. It's like having a box full of car parts. You've got all the pieces, but you don't have a car yet. It's just potential.
And then the real magic happens. Almost instantly, that long chain starts to fold. It twists and crumples and coils in on itself, all guided by the chemical attractions and repulsions between its different amino acids. It settles into this beautiful, complex, and very specific 3D shape. And let me tell you, that shape is everything. Because for a protein, its shape defines its job. So why do we care so much about this? Because these freshly folded proteins are literally the workhorses of the cell. They become the enzymes that digest your food. There are the structural bits, like collagen, that hold you together. They're the signals that let your cells talk to each other. I mean it when I say, without this constant protein construction, life, well, it just wouldn't be possible. Okay, wow, that was a whirlwind tour of the protein factory. Let's just do a quick recap to make sure all those key ideas really stuck. All right, first question. We kept talking about that 3-letter word on the mRNA blueprint, the codon. What exactly does one codon code for? Think about it for a sec. Got it? If you said one single specific amino acid, you are absolutely correct. It's the order to add one more brick to the wall. And I want to leave you with this final thought. This whole process, this incredible high-speed construction line, it's happening trillions of times a second inside you right now. But it all depends on a perfect blueprint. So, what happens when there's a typo? What happens when just one of those letters in the mRNA code gets switched? Well, that one tiny mistake can change an amino acid, which can change the final 3D shape, which can break the entire machine. And the consequences of that? Well, that's a story for another time.